natural world surges with power. It's merciless, violent, and sometimes cruel. Animals seem so regal, so perfectly suited to their environments, that we have a hard time imagining them sick. But nature can deliver some tiny, potent, and lethal surprises. Bacteria and viruses are all around us, everywhere. When they invade a vulnerable host, disease strikes.
Hundreds of seals are washing up on the coast and dying, at least 3,500 in the last 3 months, or a quarter of the estimated population of the North Sea.
I've never seen so many dead birds in one place, in one table.
A disease that spreads to animals on a large scale is an epizootic, an animal epidemic. When it strikes, how do wild animals respond? How can they protect themselves without medicine or vaccines?
Ants have been practicing social distancing for 80 million years, putting individuals in quarantine and changing their behavior whenever a parasite infiltrates their colony.
Viruses and bacteria are also part of nature. What surprising role do they play in the natural world? This is Thai National Park in Côte d'Ivoire, a well-preserved area rich in biodiversity. A team of researchers have just arrived at this lab unlike any other.
Knock, knock, knock.
Before entering the lab, the new arrivals must follow a strict protocol. 5 days of quarantine with zero contact with the outside world. This ensures the scientists won't introduce any new diseases into the forest. These researchers are studying microorganisms. Some of them can be highly infectious and contagious. They're called pathogens. Their goal is to understand the diseases of the natural world. This lab is a pioneer in the field.
We're in the heart of the virus sphere.
Léonce Quadio is a virologist, professor, and researcher in Côte d'Ivoire.
This place allows us to do different studies, because these days, all of our epidemics originate in forests like this one. So we conduct studies here, not only to identify the various pathogens in circulation, but also to determine what steps we can take to ensure they don't leave this environment.
The world is full of microorganisms, bacteria, and viruses. They're everywhere, inside us and on us, in the soil and on plants, but only a small percentage of them are actually pathogenic.
Ariane Dux is a veterinarian and an expert on animal epidemiology.
There is an incredible number of pathogens. Each animal has their own pathogens, and sometimes they spread from one individual to another. There are as many methods of transmission as there are pathogens.
There's a reason for these scientists' presence deep in the heart of this forest. It's one of the last primary tropical rainforests left in the Gulf of Guinea. It's home to an incredibly rich diversity of flora and fauna, and a UNESCO-listed World Heritage Site. This forest is also a sanctuary for an exceptional chimpanzee population. An urgent call comes in for Ariane and Léonce. One of the guides from the camp has found a dead duiker. The body of the small forest antelope was found in chimpanzee territory.
Whenever a dead animal is spotted, like in this case, we're notified as quickly as possible, and we head out nearly immediately to take samples. But there are several pathogens in the forest that can be pretty dangerous.
Before they head out into the field, they disinfect their boots and hands to minimize the risk of introducing outside pathogens. In Thai, as in other places, viruses and bacteria are everywhere, in the soil, the air, the water, and even in the bodies of the animals themselves. Most of them are harmless or even beneficial, but some of them are dangerous and contagious. To protect themselves, animals have a natural defense mechanism against these pathogens. Their immune systems, namely the animal's organs, cells, and certain molecules that protect them from outside attacks. These defense mechanisms almost always work, but sometimes a simple imbalance is enough to let a virus or a bacteria slip through the net. If it then spreads to other individuals, it marks the beginning of an epizootic disease called an emergence. From this moment on, every minute counts. Animal carcasses don't last that long in a tropical rainforest. Insects and scavengers of all sorts take care of them pretty quickly. The whole thing will be gone within 48 hours.
See over there? Behind the little log? That's where I followed the smell. I could see it was a doe.
They set up a security perimeter and take the utmost precautions during the autopsy in order to avoid potential transmission. For the moment, the researchers have no idea what the cause of death is.
We have to protect ourselves, because it could be anything. It could be Ebola, monkeypox, anthrax, or some other terrible disease we don't even know about yet. All good?
Yes.
In tropical rainforests, very few animals die of old age. There are predators and poachers, and sometimes disease. The duiker doesn't show any signs of impact or violence. The killer must have come from within.
We have very specific procedures that we follow. We take different samples depending on the condition of the dead animal. If the carcass is fresh, we analyze all the organs. But if it's been there for a while, all that will be left is a few maggots or a bit of skin.
Now they must spread out from the carcass and question their first witness, flies.
It's kind of a toss-up when we find a dead animal in the forest. We don't know if it's an isolated case or if there might be an epidemic. That's why we capture flies, because unlike us, they find all of the carcasses in the area. By analyzing them, we can determine whether they're carrying a pathogen that might have caused the animal's death.
There are three main suspects in this investigation: the anthrax bacteria, the Ebola virus, and the monkeypox virus. All three can kill their hosts very quickly. Two other populations will also be monitored closely: rodents and bats, two groups of natural reservoir species.
I suggest we set up the net here, like this.
Natural reservoirs are species that carry pathogens without showing symptoms of disease. Inside their organisms, a compromise is reached between the immune system and the disease. Bats are one example. They have developed powerful immune systems making them potential reservoirs for multiple diseases.
It's like any other investigation. We want to know who's guilty and what they're planning next. What we need to know now is if this is the only death. Was it random, or is there an epizootic amongst the wildlife?
How do pathogens conquer new territories? To answer this question, we'll head to the United States and travel back in time. It was a coincidence, one that still hasn't been explained to this day, that brought an African pathogen to the docks of New York City in 1999. In a matter of days, the megalopolis became the epicenter of one of the most widespread epizootic outbreaks in modern history.
It was shocking. It's always shocking when you see the high mortality. High mortality is very serious. It creates a lot of anxiety in the scientific community to understand what the issue is.
Antonio Garmendia is an animal virology researcher at the University of Connecticut. In 1999, he was at the center of the investigation.
In any street, in the campus here, in the university, you see dead crows on the field. When you see something like that, something is going on in nature. So this have all the features of a major outbreak. And in fact, it was a major outbreak.
The first dead crow of the season has tested positive for West Nile virus. The Wellington Dufferin Guelph Health Unit has confirmed the results. The crow was found in the Bellwood area.
Us having a service here received a number of birds, and there was an excessive number of birds, unusual. I'd never seen so many dead birds in one place, in one table.
By late summer 1999, scientists had managed to identify the virus responsible for this massacre. West Nile virus, a disease that's spread by mosquitoes.
The exact mechanism for introduction is unknown, and there are some theories. One is that infected mosquitoes could have come on a plane with that type of globalization and travel.
To this day, public health officials continue to check for the presence of West Nile virus in American mosquitoes. John Shepard is in charge of these studies in Connecticut.
When West Nile virus was identified, it was slightly unnerving because it was a new virus in a new part of the world. But it was also exciting because we were actively looking for the virus in birds and mosquitoes to see how this virus was going to affect this new ecosystem.
In John's lab, they study some 50 species of mosquitoes. One of them, Culex pipiens, the common house mosquito, has been identified as a major vector of West Nile virus. Unlike natural reservoirs, which merely preserve the pathogen in their organisms, vectors move around and spread it actively, sometimes across huge distances.
It was found at the time to be very lethargic and getting towards a lot of people. It started to bite into midair and then died on its own.
Guillermo Risati is an entomologist and the head of the autopsy department at the University of Connecticut.
The most important feature of this disease is how in a short period of time spread across this nation. So basically the vectors were present here. They were able to transmit the disease or to transmit the virus both to birds and to animals and eventually to humans.
On the African continent, animal immune systems have adapted to the presence of the West Nile virus. The virus has become what we call epizootic. It's part of the environment and emerges only occasionally without any major consequences, like the flu for humans in winter. But when the Nile virus reached New York City in 1999, the contaminated individuals had never been exposed to it before. Every new animal that it encountered was a potential victim. As the virus spread, it left death in its wake. Thousands of small mammals and hundreds of thousands of birds died in the first few years. In 2002, it spread to mustangs and domesticated horses, killing off 30% of the population in the Midwest. The virus also affected humans, causing thousands of deaths. From a simple emergence in New York in 1999, West Nile virus spread across the entire continent in just a few years.
No external parasites, the eyes are sunken in, and nothing in the mouth.
Mm-hmm. We're going to keep looking for West Nile virus for a long time. So time to time we will have these situations that we will see outbreaks of the disease. So we need to monitor for that. We need to see for the emergence of new variants of the virus. We need to be very proactive in that sense. That's why we do that.
In Thai, no one knows if the duiker they found might be the beginning of a new outbreak. Ever since its carcass was found, the forest has been under close surveillance. The first witnesses have been captured. Bats are potential carriers of Ebola. The last time Ebola broke out in the Thai forest in the 1990s, the chimpanzee population was nearly wiped out. We're still not sure how the Ebola virus is transmitted. What we do know is that it's present in bodily fluids, like bat saliva.
There generally isn't any direct contact. The epizootic spreads through fruit eaten by a bat. If the bat happens to be infected with a pathogen, then obviously that fruit will also contain that pathogen. Then if an animal, like a deer, comes along and eats the rest of the fruit, the animal will be indirectly infected with the same pathogen.
The last witness they interrogate tonight is a wood rat, from which they take a mucus sample. Then they carefully release it back into its habitat. All of these pieces of evidence will be stored and analyzed as soon as possible.
Pathogens are often transmitted from one species to another. In some cases, this can have terrible consequences. That's why it's so important that we go check on the chimpanzees to make sure they're all still there and that they're healthy and acting normally.
As they wait for the results of the analysis, the tension is palpable. Tomorrow, Ariane and Roman will go out to observe the chimpanzees in their territory. That's where the dead duiker was found. There are nearly 900 chimpanzees living here, one of the largest populations in West Africa. Around 100 of them have been monitored by scientists for the last 40 years. Roman Wittig is a primatologist and the director of the Thai Chimpanzee Project. The scientists know exactly where the nests are. After a 2-hour walk, the great apes finally come into view.
There's Gemma up there. She's the new female.
Okay, how long has she been in the group?
Oh, it's about half a year, I would say. But she has still, you know, the full swelling. And the males are still crazy. And the females hate her. But it's good that she is with Mister, I think. Mister is sitting up there. Mm-hmm. Yeah.
Ariane has spotted a clue that must be collected immediately.
Feces are very important to us because we can collect feces without bothering the animals. It's minimally invasive, and we don't need to put the animals to sleep or anything like that. It's very simple, and it allows us to detect nearly any pathogen. We have the largest collection of great ape feces in the world at our institute in Greifswald, Germany.
By analyzing this sample, they will also gain insight into the chimpanzee's recent diet. If the disease is present, there's a good chance they will have changed their diet. They may have even found a remedy in the forest.
In Thai, we know that chimpanzees, during a certain season are indeed, using a specific type of leaf that has little hooks, on one side of the leaf, and then they roll the leaf with their tongue and they swallow it entirely. They are potentially trying to collect, the eggs of intestinal parasites on there.
Self-medication has been observed among great apes by generations of researchers and is the origin of the field known as zoopharmacognosy, or the study of how animals self-medicate. Jean-Marc Dubost is an ethnobiologist who studies the elephants of Laos. Over the course of his many travels, he has documented the pharmacopoeia of pachyderms. This is the Elephant Conservation Center in Nam Pui National Park, Laos.
We've found examples all throughout the animal kingdom, whether in insects, birds, or mammals, of animals displaying self-medicating behaviors. Elephants are no exception, of course. Boiban, that you can see here, is a good example. He had a suppurating wound on his left hip that got infected. We had been treating him for a year using conventional treatments, which means we were disinfecting his wound, cleaning it, and giving him antibiotics and anti-inflammatories. None of it had much of an effect. He was starting to fail. He had lost a lot of weight. Elephant drivers believe that if an elephant is sick, the best thing to do is release it into the forest for 2 or 3 weeks. They say the elephant will find what it needs to get better. But the vet said that if we released him into the wild, it was as good as leaving him to die. Considering his quality of life at the time, he was doomed either way. I saw him a month later. I went into the forest with his elephant driver, and that's when we filmed this footage.
Jean-Marc Dubost found Boiban in the forest. Against all expectations, the animal was in perfect health. After being left to his own devices in nature for a month, he had recovered his strength and cured his wound. As ultimate proof of his recovery, he offered the researcher and his elephant driver a moving spectacle.
He spent an hour taking apart an entire tree. You can see him eating. He must have eaten 4 or 5 kilograms.
Throughout their observation, Boisban showed no signs of weakness or fragility in the hip. By eating plants from the forest, he had completely healed himself.
It's interesting to see that kind of symbiosis in nature, in all sorts of plant and animal species. It occurs on every level.
Jean-Marc Dubos has identified 20 different plants that elephants use to heal themselves. But even before they fall ill, animals also practice preventive medicine. Melliferous bees, for example, build their hives using antimicrobial materials, most notably propolis, which they gather from the buds of certain trees. Blue tits of the Mediterranean basin use butterfly lavender, round leaf mint, and immortal flowers from Italy to build their nests. The antiseptic properties of these plants prevent the spread of certain bacteria. Other species isolate themselves from the group to prevent the spread of disease. Bullfrog tadpoles distance themselves from contaminated individuals before they even show any symptoms. Spiny lobsters isolate themselves when they're infected by a virus. They retire to a sandbank, where they're at the mercy of predators. Instinctively, they prioritize the survival of the species over their own. When there's a threat of illness, chimpanzees, which are traditionally highly social animals, change their behavior as well. They avoid certain areas and sometimes split up into smaller groups. Within their nests, they touch each other less and keep more distance between them. But for now, Ariane and Roman are reassured to see that the group is still together and acting calm. Like the great apes, most social species change their behavior in the case of disease. That's also true for a group of species that have evolved very successfully, that continue to be omnipresent around the entire surface of the globe, and which still hold many secrets. Laurent Keller is one of the world's leading myrmecologists. For the last 30 years, he has been studying ant behavior at the University of Lausanne.
We've identified 14,000 species of ants, and there are still tens of thousands waiting to be discovered. Why have they been so ecologically successful? Because they're social and because they have a highly complex society with a division of labor. We learn something new about them every day.
Ants are everywhere and have been on the surface of our planet for millions of years. Yet the way they live in densely packed anthills should make them particularly susceptible to the spread of pathogens. Then how do they fight off disease?
In order to study ant behavior, scientists used to paint them colors, but that didn't tell us very much, so we decided we needed something more sophisticated.
The first step is to put the ant to sleep. When subjected to temperatures below 0 degrees Celsius, these insects go into a state of rest, where they can be equipped with a special accessory.
We came up with the idea of gluing barcodes onto the worker ants. Alexandra, how many pictures are we taking per second?
We're at 8 at the moment.
Let's follow them for 2 weeks.
Okay, 2 weeks is fine, no problem.
We use cameras to take several photos per second, which tells us the location and orientation of each individual ant. We can follow them like that for weeks. We know exactly what each individual is doing and who's interacting with who through their antenna. That makes it possible for us to follow each ant individually and recreate the social network of the colony as a whole. We can reconstruct a sort of Facebook for ants, but a much more detailed Facebook than the human version.
After multiple tries, the system is finally ready. The experiment can begin. Over the course of several weeks, the researchers infect a batch of ants they're tracking with a fatal, transmissible fungal spore.
We discovered two things. The first is that the location of each individual in a colony is closely linked to their job. For example, ants who go out foraging for food stay close to the entrance of the nest, while the nurses who take care of the babies stay close to the babies. They never approach the entrance to the nest, which means there's very little interaction between foragers and nurses.
In other words, ants naturally practice a form of isolation within their own society.
The second thing we found is that the way the ants interact with one another changed when we introduced the parasite. The amount of separation increased, thereby lowering the chances of transmitting the parasite to the most important individuals in a colony, namely the queen and the babies. And the sick individuals infected with these spores isolate themselves. They kind of put themselves into quarantine. If an individual dies of the infection, the other ants will put them in the cemetery to keep the parasite from spreading.
The discoveries by Laurent Keller and his team are fascinating. Ants seem to have always practiced certain behaviors that protect the colony as a whole.
When COVID first appeared, it took us a long time to realize we had to stay several meters away from one another rather than getting too close. So as humans, we're still learning things that ants have known for 80 million years, which are the ideas of social distancing, putting individuals in quarantine and changing their behavior when a parasite is introduced in a colony.
Ariane and Leonce have returned to camp, reassured by the chimpanzee's behavior. They now get back to analyzing the samples taken from the carcass. Their priority is to identify the pathogen that caused the animal's death in order to prevent a potential epizootic outbreak. Despite the rustic appearance of the wilderness lab, it allows the scientists to conduct critical analyses.
The samples were tested for two different pathogens, the Ebola virus and the anthrax bacteria, which are the two most dangerous pathogens in the forest. Fortunately, we were able to rule out Ebola, but after conducting some DNA sequencing and doing a PCR test on the samples, we were able to confirm that the cause of death for this duiker was anthrax.
Anthrax is an infectious disease caused by a bacteria known as the coal bacillus. There is little to no chance of survival after infection. Anthrax is present throughout the Thai forest and kills primarily herbivores, but also sometimes chimpanzees. Victims are contaminated by ingesting bacterial spores present in soil, on plants, or in fruit that has fallen onto the ground. These spores germinate inside the animals' bodies and turn into bacilli that produce toxins that kill them. As the carcass breaks down, the bacteria produce new spores that can survive in the soil for decades or even centuries.
Anthrax can infect nearly any animal in the forest, except for certain carnivores. But it's particularly dangerous to our chimpanzees. It has killed chimpanzees nearly every year for the past several decades.
The bacteria responsible for killing the duiker has been identified. But the investigation isn't over yet. They still have to find the specific strain, which is to say the genetic family of the bacillus they've recognized. The goal is to know whether it's a new variant or one the researchers already know about.
We always collect 2 samples. The second sample is transferred directly to Germany to be archived and also to confirm our findings.
Pathogens are constantly mutating and adapting, just like the animal's immune systems. This continuous evolutionary competition presents a challenge to researchers studying the coevolution mechanisms of pathogens and their hosts. In the Kattegat Strait in Denmark, this phenomenon is tragically illustrated by the evolution of seal populations. They've been dealing with extremely violent epizootic diseases for decades.
The first outbreak happened in 1988, and the second one happened in 2002. So if we would follow that pattern, then 14 years later, we would have expected a new outbreak. But nature is not always so consistent.
Iben Stockholm is an ecologist at the University of Copenhagen. Today, she is monitoring seal populations in the Kattegat Strait, where the virus is particularly devastating.
Hundreds of seals are washing up on the coast and dying, at least 3,500 in the last 3 months, or a quarter of the estimated population of the North Sea.
Rune Dietz is a professor and ecologist at Aarhus University. He was one of the witnesses to the terrible first wave.
Such a mass mortality is one of the largest mass mortalities we have observed among animal population.
In 1988, 60% of the population, meaning 30,000 seals in the Kattegat Strait, were killed. In 2002, the virus made a comeback, killing 40% of the population. At first, no one knew what virus was responsible for these deaths.
It was a novel virus. Nobody had ever seen it before. It had ever not been sequenced before. So we didn't know that much about it. We didn't know where it came from.
The researchers hence sequenced the RNA of the virus, which is to say they analyzed its genetic makeup. That's how they were able to identify the guilty party.
At the end of the day, they figured out that the pathogen killing all of these seals were in fact a pathogen that was related to another pathogen we already knew, which is called canine distemper virus. It's a very dangerous virus for dogs to get, and that is the closest relative of porcine distemper virus, which is the seal virus that they found there.
Like with the dead duiker in the Thai forest, knowing the name of the pathogen wasn't enough. They still had to figure out what strain it was, and also understand how it had gotten to the Kattegat Strait.
Because then we don't catch anything.
And the most risky thing today is to not get caught in the net as it goes out.
Actually, I had collected samples in the Arctic region prior to the first outbreak in '88. So I went into my freezer and took samples. And then we found actually the distemper virus from the Arctic region. And that was part of the proof that this disease came from the Arctic region and invaded the European waters.
Right before the epizootic in 1988, there was a huge migration of harp seals, which are from the Arctic. And due to overfishing, the capelin stock crashed in 1987. So there was not enough food for the harp seals to eat, and therefore they were looking for other places to get their food.
In Greenland, seals have been confronted with the virus several times over the last few decades. Over time, their immune systems have adapted to the disease, demonstrating once again the phenomenon of co-evolution between hosts and pathogens. But Arctic seals, which are asymptomatic carriers of distemper, ended up acting as a vector to transmit the disease. In Kattegat, the strongest of the Danish seals survived by developing antibodies. Little by little, they achieved herd immunity. The population slowly grew back, and over the years, the virus disappeared.
The thing is that we can see that the seals that were infected back in 2002, they are old now or dead, and a new generation has been made, and then they don't have any antibodies anymore.
Scientists are unable to explain this reduction in antibodies over time. But today, they can monitor it by taking samples. While Arctic seals seem to have developed a long-standing immune response to the disease, the seals of the Kattegat Strait find themselves at the mercy of a new epizootic wave.
So we are expecting that there will come a new outbreak. And now we see climate changes, and maybe some places will be overfished again. And when this happens, the animals, they will begin to migrate. And then maybe we will see a new outbreak within the coming years.
The scientists remain on alert. As part of a European program, Eben and Room's findings will be cross-referenced with those of their British and German counterparts. We meet back up with Ariane and Leonce in northern Germany. They have followed the samples from the Diker. They're now in the city of Greifswald, one of Europe's most important university hubs. One of the local labs is in the Helmholtz Institute for One Health, directed by Fabian Lendertz, who's in charge of some of the research being done in the Thai forest. His goal is to better understand the interactions between living beings in order to come up with a global approach to health.
We created a platform, which is a One Health surveillance, looking at the environment, at human health and the animal health, including biodiversity. It's like a little weather station where you're trying to monitor the climate and the weather over many years. It doesn't help to look at this for 1 year. You need that for 20, 50, 100 years if we are lucky.
The co-evolution of pathogens and their hosts is one of the main areas of study here.
We sequence the entire genome of every strain of anthrax that comes into our lab, and of nearly every pathogen, if we can. That helps us do pathogenic analysis, which means determining the genealogical trees of the pathogens we encounter. That, in turn, tells us if we are dealing with new strains and if they all behave the same way.
Leonce starts the complete DNA sequencing of the bacteria taken from the Diker carcass. He hopes to find the origin of this anthrax in the bacteria's phylogenetic tree.
Here, highlighted by the star, we have our sequence from the Diker.
To build this tree, scientists have cataloged samples for 20 years. They start by noting on which species the anthrax was collected. Then they connect them to other anthrax bacteria that had previously been analyzed based on their genetic similarities. If they discover a new, isolated branch on the tree, it means they have identified a new variant.
As you can see, it's a heterogeneous mix, which means that the bacteria isolated on the flies, chimpanzees, monkeys, and other animals are all mixed up together. Therefore, there isn't one particular group infecting only chimpanzees, for example, but all of these strains of bacteria are infecting multiple species.
Okay, great. And I think as far as we know from the other clusters we looked at before, that you can find the same type 10 years apart. So there doesn't seem to be a temporal cluster. So it's really some kind of parallel circulation, probably.
Yeah. I mean, there's a chimp in the tree, and the last chimp that died of anthrax should be at least a year ago, no?
Okay, yeah, I see. Okay, very nice. Mission accomplished.
The guilty party is finally identified, but will never be arrested. In nature, pathogens always go free.
We classically think of pathogens as something nasty which makes us sick and kills us, which is true or can be true. But this is because we're focused on ourselves. But if we look at the bigger picture, pathogens are part of the natural balance we see worldwide.
Nature has always dealt with disease. In fact, pathogens are a part of the natural balance. One species perfectly illustrates this natural balance in how it responds to disease. It's an example of extraordinary resilience. At the foot of the Altai, in far western Mongolia, a team from the WWF is attempting to capture one of these animals, the second fastest on the planet. The saiga antelope.
These brave animals have been living in this arid climate since the dawn of time and still do today.
Baljinyam Batsaikan is a veterinarian and WWF supervisor. Today, his goal is to fit a saiga with a GPS collar so they can better monitor the evolution of the population and their exceptional ability to adapt to disease. But to that end, they have to keep pace. In the immense rocky steppes, nothing could be more difficult.
You see the two saigas over there? A viral epidemic known as ovine rinderpest spread in November 2017 when the antelopes traditionally gather before their mating season. I was working with the saigas when the epidemic struck. It was terrible. They couldn't walk. You could see saliva, mucus, phlegm, and tears flowing from their nostrils and their mouths. They couldn't stand. It was heartbreaking to watch. The antelope died within 3 days of being infected. It's a fast and dangerous epizootic disease.
At the time, the virus killed 70% of the population in just a few weeks. It had probably come from the herds of livestock animals. Saigas died by the thousands. However, their population has not stopped growing since.
Going from a population of 3,500 to 10,000 in just 4 years is just extraordinary. That will never cease to amaze me.
The numbers are simply astounding. In just a few years, the saiga population has tripled. Quite the exploit for a ruminating mammal.
We noticed that with these wild animals, when the population decreases, they tend to start having twins to bring their numbers back up. We didn't know about them before because they had never caught an infectious disease.
The scientists discovered that after an epizootic outbreak, saigas reached sexual maturity at a younger age, thus accelerating their rate of reproduction. Moreover, the gestation period became longer, which meant the females gave birth to stronger, sturdier calves. Thanks to this exceptional demographic rebound, the saigas counteracted the devastation caused by the disease in a matter of years. But they aren't the only ones in the animal kingdom. In 2007, the reproduction rate of white-tailed deer adjusted in response to an epizootic outbreak. Females gave birth to larger litters, thus compensating for the higher mortality rate among young deer. Another study showed that the percentage of reproductive females among red foxes went up significantly after a rabies epidemic. Viruses, bacteria, and wild animals have been cohabitating and co-evolving on our planet's surface since the dawn of time.
Pathogens are always evolving. They're never stagnant. They mutate and adapt to new factors. And then the animals adapt in turn. It's a never-ending arms race between pathogens and hosts. They're constantly changing.
In this strange eternal relationship, a dynamic balance is always reached. Diseases in the wild manifest in many ways and have multiple causes. But more and more often, they're a result of humans and our impact on ecosystems.
Humans have destroyed the natural balance in many, many ways. And we humans are so many on this earth now, and with such a high mobility, that we bring pathogens to the different parts of the world. So we are a very important reservoir for diseases for animals as well. The question is, how can we find a new balance with nature? And there's a way that all species have a chance to survive on this planet.